So hi everyone, I hope you're all well and you're not too stressed from all of the assignments that you have been given in recent weeks. Today I've been tasked with the job of leading the next session for your applied methods part of the course. And today what I'm going to talk about is inflammatory biomarkers and specifically talk about protein biomarkers and how you can detect proteins in inflammatory diseases. So if we make a start on today's session, so here's just the contents in detail for today's session. So what we're going to do is talk about inflammatory biomarkers. So what are they? I'll provide some examples of these biomarkers that you can detect in certain oral diseases. And then I'll provide examples of clinical samples used for detecting biomarkers. And then in the second part of the session, we'll go on to talk about methods that are used for, or that are commonly used for detecting inflammatory biomarkers. Specifically, we'll talk about ELISAs in quite a bit of detail because this is going to be the methodology that we are essentially using for the data analysis part of this session. And I'll record that in the next, I'll record that part of the lecture next. And we'll also talk about multiplex assays, so specifically flow cytometry has been used to basically detect proteins in clinical samples. I'll touch upon briefly protein immunofluorescence, immunohistochemistry, and then I'll finish off by talking about next generation platform technologies. That's kind of a term that I've used here, just sort of exciting novel methods that are now being used within or you've been used within laboratories for detection and quantification of proteins. And specifically, I'm going to talk about Olink. The first part is going to be very simple. So what are inflammatory biomarkers? Well, the level of inflammation within a given patient can be estimated by measuring circulating substances. And one of these circulating substances will be proteins. And these proteins are released as a cause or a consequence of an inflammatory response. And these inflammatory responses are basically the immune system responding to certain microbes, for example. And obviously, if we think about the oral cavity, there's many different types of oral diseases that can have some role or involve some role of the immune system. Examples of these proteins that can be produced during this process are C-reactive protein, inflammatory mediators such as cytokines and chemokines. So these cytokines and chemokines are signaling molecules that instruct cells, immune cells, what to do. And then there's other host proteins as well, depending on where you're talking about within the human body. These proteins can also be produced by different types of immune cells and also other host cells. And good examples of these will be enzymes or antimicrobial peptides, which function in trying to prevent microbes from attaching to the host and thereby invading the host and causing infection. And these biomarkers can be detected using a variety of methods, and we'll talk about those in today's session. So just to go into a bit more detail on specific biomarkers that are commonly assessed or quantified within clinical samples within the human body. And the first type is C-reactive protein. And this is something I think we've encountered in journal club sessions and papers from journal club. And C-reactive protein is just a general generic marker for systemic inflammation. So you find it circulating within the blood in a certain inflammatory disease or a certain inflammatory response within a given disease. It's produced by liver cells and specifically these hepatocytes. So these hepatocytes are basically just specialized cells of the liver that produce C-reactive protein. Production of C-reactive protein is controlled by immune complexes as well as cytokines such as interleukin-6. Also controls the production of C-reactive protein. And basically it's these immune complexes or these cytokines that can form during an immune response. So I've talked a bit about antibody and antigen immune complexes when I gave the vaccines lecture, which hopefully you will have watched by now. And once I upload this presentation to Moodle. So immune complexes where you have an antibody recognizing an antigen these complexes can drive the production of C-reactive protein within liver cells. These liver cells will produce this protein, and this protein, essentially its main function is as an opsonin. And an opsonin is ultimately a fancy term for a protein that can coat a certain microbe or tag a certain microbe so that the immune system can target it and remove it. And one of the ways that it does this is by basically activating the complement cascade. So the complement pathway is part of the innate immune response. And the primary role of the complement cascade is to produce these membrane attack complexes that form in the surface or on the surface of microbial cells. So here in the electron microscope image, what you can see is a rod-shaped bacteria. You zoom in really high magnification, you can see these pore-like structures forming, this membrane attack complex forming within the microbe or on the microbe surface. And this ultimately leads to lysis of the cell. So C-reactive protein is really important in driving this process, which is essential in trying to remove an infectious agent from circulation. So there are other types of inflammatory biomarkers. So the most common ones are cytokines and chemokines. So as I've said, these are small signaling molecules that are produced by different immune cells within the body. You have pro-inflammatory cytokines and chemokines and anti-inflammatory proteins as well. And it very much depends on the level of these given cytokines or chemokines. It can actually dictate and inform you at what level of inflammation is occurring within a given patient, for example. So for instance, if you have a clinical sample, let's say if you took the blood of someone and found a high level of IL-8 or TNF alpha within their blood, then of course, then you could argue that they have a high level of inflammation at somewhere within the human body. And it's the complete inverse or converse when you think about anti-inflammatory markers as well. So if there's a high level of interleukin 4 or interleukin 10, for example, then it's kind of an indicator that there is no inflammation going on within a given patient. And when you're able to quantify these and detect these within a clinical sample, this will give you an indicator of a level of inflammation within a given patient. Antimicrobial peptides are a different type. So these function as neutralizing agents against microbes. The main function is to destroy or disrupt membrane integrity. The formation of pores can also be driven by certain antimicrobial peptides. They can also cause intracellular toxicity as well, not only in bacteria, but other microbes as well, such as fungi and parasites and also viruses. And then there's also other proteins and enzymes specifically produced at things like mucosal surfaces. And of course, the oral epithelium, the gingival epithelium is a great example of a mucosal surface. So things like proteins and enzymes are often found in things like saliva. And these function to try and prevent microbes from attaching and invading the host. So if we move on and specifically talk about inflammatory biomarkers within the oral cavity, and I'll provide some examples of these and also examples of clinical samples that you can possibly use when you are trying to detect biomarkers. So when you are, let's say, coming up with a research proposal, which is of course part of the assignment, you need to take into account multiple things. So it's very much dependent on the disease. The disease will dictate what biomarker you want to assess. So for instance, if you want to assess periodontal disease or oral cancer, then of course you're going to have to pick slightly different biomarkers because there's going to be slightly different biomarkers that play a role in the pathogenesis of these diseases. And it's going to be the same when you maybe think about dental caries, which doesn't have that much of an inflammatory response associated to it. Then of course you may have to pick certain things like enzymes or proteins that you find in saliva, for example, that may prevent organisms such as Streptococcus mutans from attaching to the tooth surface and thereby driving this demineralization of the tooth. Or they may be proteins, for example, that can actually neutralize pH that you find in saliva. So the take-home message here is it very much depends on the disease, and that will dictate what biomarker you can select. And secondly, it's also very dependent on the clinical sample that you want to take. So for example, if you was taking the plaque, whether it be the sub or the supragingival plaque, then of course you're only going to really be able to assess the microbial composition or the microbiome of the plaque. You're not going to be able to look at anything to do with the immune system, which is fine if that's the research proposal you want to come up with. Then of course you can look at the last two sessions that Professor Ramage, Tracy, and also Mark delivered. And if you want to go down the microbiological side of things, then plaque would be an excellent clinical sample. On the other hand, if you want to go down the immunological route with your research proposal, then things like saliva or GCF would be excellent examples. The GCF specifically would be chosen if you were looking at periodontal disease. Saliva, on the other hand, would be maybe chosen if you're looking at generic markers of oral cancer, for example. The second thing is, or what you could take is biopsies. So we'll talk about immunofluorescence, staining and immunohistochemistry later on in the second part of the talk. But hard and soft tissue biopsies are also feasible options as well. So for instance, if you were looking at periodontal disease, you could take soft tissue of the gingiva and a biopsy of that. For hard tissue, you could take things like, or disease-wise, this would be something like dental caries or maybe endodontic infections. And then finally, systemically, of course, you can take the blood and you can assess the systemic response within a given patient and see whether an oral disease is affecting the systemic health of a given individual. So when we are talking about biomarkers in the oral cavity, what could you look for? Well, the epithelial barriers within the oral mucosa produce several different proteins. So these could be antimicrobial peptides, which we've already talked about, cytokines, chemokines, but also there's other types as well. So there's enzymes such as lysozyme, immunoglobulins, so secretory IgA is a type of antibody. Again, I probably covered a bit of that in my vaccines lecture, I believe. You also have other types of proteins, things like lactoferrin and cystatins as well, that play a role within the oral cavity. But generally speaking, if you are looking at biomarkers within the oral cavity, on the inflammatory side of things, then cytokines, chemokines are the main types of protein biomarkers that you would look for. And specifically, if you were looking in systemic health, then CRP is probably the most generic protein that you would find circulating within the blood of a patient with a certain inflammatory disease. But this is just to make you aware that there are other types of proteins that you could potentially look at, but you'd have to obviously postulate and come up with a certain research question, which would then justify you maybe picking a slightly different protein. But the most common ones are cytokines, chemokines, and then systemically it would be CRP. So if you move on to the second part of the lecture, I'll just take a drink. Move on to the second part of the lecture. And in the second part, I'm going to talk about the methodologies that you can use to detect inflammatory biomarkers. And the first part of the talk will be on ELISAs. And there's different types of ELISAs. And ultimately, ELISAs stand for enzyme-linked immunosorbent assays. So ELISA, so ELISAs. There's multiple different types of ELISAs, but they all have a very similar principle. And that principle is that you have antibodies that are specific for a given antigen. And in this case, the antigen would be a protein biomarker of a clinical sample or in a clinical sample. And basically the antibody that is specific for that protein will bind to that protein, and you're able to detect that protein within the assay. Because of the to do with the interest of time, I don't have enough time to go through each of the individual ELISAs and the protocol attached to them. So the one that we're most interested in, and the one that we mostly use in the dental school in Glasgow, in the research laboratories, is the sandwich ELISA. And in the next few slides, I'm going to go into detail and talk about what is a sandwich ELISA and what are the principles behind this type of ELISA. But the take-home message here is that there's different types, but they all use a very similar principle. So as I've said, ELISAs are commercially available, and the most commonly commercially available ELISA is the sandwich ELISA, and this is used specifically to detect proteins such as cytokines and chemokines. And they can be used for different types of clinical samples. So we, over the last few years, have some nice data showing that you can detect cytokines and chemokines in saliva, in GCF, and also systemically in blood and serum. So these are the main components that make up an ELISA kit that you buy from a commercial, from a company. And this on the right-hand side just basically shows the biological principle, or a schematic highlighting the principle of an ELISA. And what I'll do in the next few slides is go through and specifically talk about each of the individual components and what they do. And I actually have a video in the next slide that should hopefully help to describe the process. So we have microtiter plates. So these will be specially made microtiter plates that are that have been specially created by the company so that they can be used only for ELISAs. So these aren't your generic microtiter plates. They'll be coated in a certain way to basically promote protein binding of whether it be your clinical sample or whether it be, in this case, the capture antibody. The capture antibody, on the other hand, sometimes this comes with coating buffer. This is ultimately the antibody that you will bind, or that you will add first to your microtiter plate, and this will bind to the bottom of the microtiter plate well. You have protein standards, and we'll talk about that later on in the session. And of course, you'll have your clinical sample, whatever that may be. And these protein standards will, of course, be known concentrations of your protein of interest. And then in terms of your clinical samples, this will be a protein of unknown concentration. And specifically what happens is the protein will attach to the antigen binding site of the antibody, whereas the constant region of the antibody will attach to the bottom of the microtiter plate well. You then have a detection antibody. The detection antibody will then bind to the protein if it is there in the well. And then you will have possibly a secondary antibody, which is conjugated to an enzyme. Or it may not be an antibody, it may just be a protein, but either way, it is conjugated to an enzyme. And this antibody will then, or this protein will recognize the detection antibody. And then basically, if that protein of interest is present within the well, then you will have the enzyme present within the well, following the addition of the conjugated enzyme. You then have a colorimetric substance. So this could be basically any type of substance that can be used within this reaction so that the enzyme will cleave that substrate into a colored solution. So generally speaking, these solutions are clear or colorless, and in the presence of the enzyme within a given well of a microtiter plate, the substance will be cleaved into a colored solution, and in most cases, the solution would be blue. You have wash buffers. So wash buffers are really important in ELISAs. Every single step that you do, and we'll talk about this in the video in the next slide, every single step that you do in an ELISA, you have to wash following incubation. And this is ultimately so important because you need to get rid of any unbound antibody or any unattached protein. And then finally, you have stop solution. So this is generally a weak acid that is sometimes supplied with a kit. Sometimes you can just make it up yourself. So it would be a weak acid such as hydrochloric acid. And the primary aim of this is to stop the reaction. So this substrate cleaving will constantly happen if you don't stop the reaction. And generally speaking, after about 15 to 20 minutes, you'll stop the reaction. If you don't, then you will get oversaturation of the color development. And basically all of the wells in your microtiter plate will eventually go a dark blue color. And therefore you need to stop it at a specific time. So hopefully that makes a bit of sense. This is essentially a video on YouTube, and I will upload the link to the video when I upload this presentation to Moodle as well, so you can go away and watch that in your own time. Instead of me being in the lab and recording the ELISA protocols, I've cheated a bit and I've found a video online on YouTube that basically summarizes the ELISA protocol within a few minutes. I think it's like a 3-minute long video. Unlike some of the techniques you've already learned from Mark and Tracy and Gordon, ELISAs take a long time, so they can take anywhere from 4 to 8 hours to run, and the vast majority of them is just incubation times. So to save you from having to sit there and watch me do an ELISA for 8 hours, I've basically just cheated and found this YouTube video, which essentially summarizes the protocol in 3 minutes. So you are very welcome for that. So hopefully this video will play, and what I will do is I'll mute the video. I don't think there's no talking on the video, but I will mute the music and I will try and talk through the protocol, if it plays, that is. Which it is not playing, which is a shame. Yes, it's playing. Right, that's good. So the video is playing now. So what I'll do is I'll talk through it. So this is the sandwich ELISA protocol. Generally speaking, you always need to bring up your reagents to room temperature. So this will be either your standards or your samples that you may have frozen. Of course, you need to thaw them and bring them up to room temperature. This is essentially what all of the reagents look like. So you have things like your stop solutions, your enzyme, your substrate, your detection antibody, your standards, your assay buffers, your wash buffers as well. And also you have your microtiter plate. So the microtiter plate will need to be coated with detection antibody, with capture antibody, generally speaking overnight, but you can also buy in pre-coated ELISA plates, and that is essentially what is being shown in this video here. So this is a pre-coated ELISA plate where the ELISA plate specifically already has the capture antibody coated in each well. So the first step is to prepare your reagents. The first step is to make your standards. So the standard comes as a powder. Usually you need to reconstitute that powder with a given volume of the buffer. Make sure it's properly mixed and then do serial dilutions. And we'll talk about the serial dilutions in the next slide. And then after the capture antibody has, of course, been given time to attach to the bottom of the well, then you need to do the first wash step. And this is the first wash step here. And they use a fancy machine. We don't have this technology at the Glasgow Dental School. We do this by hand, basically just using a squirty bottle. But basically it's just wash buffer. The primary aim of that is to try and get rid of any unbound capture antibody. You don't want any unbound capture antibody sitting in the bottom of the well unless it's already attached to the bottom of the well. So they use a fancy machine to wash the plates. And then after the first wash step, they add what is known as assay buffer. So the protocols will vary depending on the commercial kit that you buy. So they'll add the assay buffer here before then adding their samples. So they'll either add their standards at the varying concentrations that they've serial diluted, or samples of the clinical samples of unknown protein concentration. The following addition of the samples, they'll seal the plate with a plate sealer, and then they'll basically incubate for up to 2 hours at room temperature. You can also incubate it in the fridge overnight. The next step is to discard the contents and then wash the plates again. Generally speaking, you always wash ELISA plates around 4 or 5 times to properly get rid of any excess substances. Following that wash step, you then add the detection antibody. So this is this one here. So if the protein is present, or the protein of interest is present within your clinical sample, of course, within the standards, then the detection antibody will bind to it. And to do this, you have to leave it incubating again for around an hour to 2 hours, depending on the kit. You discard the contents as normal, and then you wash the plate again with wash buffer. So many wash steps, many different times. So it can be quite a time-consuming experiment or protocol. So the next thing to add is the avidin horseradish peroxidase solution. So this is basically just the enzyme attached to a certain protein. This protein here is highlighted in this blue image here. And basically this is the enzyme that will attach to the detection antibody if it's present within the well. And you have to incubate it again and allow this attachment to occur. You discard the contents again, and then you wash the plates again with wash buffer. So they suggest to wash a bit more excessively in this final step to get rid of any non-bound enzyme. And then finally, it's essentially the adding of the substrate. So this is substrate being added. You can see that it's clear, colorless solution. You then incubate in the dark. Usually we use tin foil that you use for your cooking of maybe your chicken or your turkey. And basically, because it's a colorimetric change that you see, you need to make sure that there's no light present that can drive this process. Because if you leave it in sunlight, for example, if you've got maybe quite a bright room, or if the lights are quite strong, then this color change reaction will occur in all of the wells. So you need to make sure you incubate it in the dark. And in the presence of the protein, what will happen is you get the color change of a colorless solution turning into a blue color. And then once you've left that to incubate for anywhere from between 15 and 30 minutes, depending on the kit, you need to be able to stop the reaction. And to do this, they use the stop solution. As I've said, this is generally just a weak acid. And as you can see here, the blue color is turning into a yellow color. You then read the absorbance at 450 nanometers immediately. And this is essentially a look at what data you will get back. So you'll be able to get a standard curve, which will be on the x-axis will be the concentration of the standards, of the serially diluted standards. On the y-axis, you will have the absorbance values. And you'll basically have a standard curve, which you will be able to use to essentially calculate what concentrations of proteins you have in your clinical sample. So that is, as I say, that's just a very whistle-stop tour of an ELISA. This protocol can take anywhere from between 4 and 8 hours because of the many wash steps and also the many incubation steps as well. But that should give you an idea of the very basic principles of an ELISA. So as I've said, standards are really important. Generation of a standard curve will allow you to calculate the concentrations of your protein of interest. This is essentially a plate, and this is actually going to be the plate that we use to work through as an example, or the absorbance values from this plate will be the example for the data analysis part of this session. And this is essentially what it will look like. So you have in columns 1 and 2, this is your standard. So you can see a nice dark blue color at the top. And that blue color, there's a gradient of blue essentially going down from dark blue all the way down to quite a very light blue color. And then all the rest are your clinical samples. So you want to then be able to use this standard curve to determine the concentration of the protein within your clinical samples. And in terms of doing standard curves, in terms of generating a standard curve, you have to serially dilute your standards. So generally, you'll have a very concentrated amount of protein provided to you within the commercially available kit. You will then need to dilute this quite excessively before then you dilute it twofold dilutions to basically produce a standard curve. And generally the concentrations that we use and the ones that we'll use for the example are these here. So you'll have 2000 picograms per mil all the way down to around 32 picograms per mil. And also you should always include a background control. So this will basically be a blank. There'll be no sample in this. And in this example here, it's quite difficult to actually make out, but this H1 and H2 is basically a blank. So this contains no sample. This will just give you a background reading of the absorbance at that wavelength. And as you saw in the video, briefly, this is essentially the data that you will get back. So in our example, in the next recording that I'm going to do, we're looking for IL-8. So this is concentration of IL-8 versus absorbance at 450 nanometers. And basically what you'll see is this kind of standard curve, which looks like a bit like an S-shaped because it's a sigmoidal curve. It's not a linear relationship between concentration and absorbance. And the standard curve will allow you to determine the unknown concentration of the protein in your clinical sample. So if we have an example here, if your absorbance at 450 was 0.75, so here, if you worked across and used the standard curve, what you would be able to do is identify that this is around 380 picograms per mil of IL-8. And you'll be able to do this using GraphPad Prism. And as I say, we'll talk through that in the next session. In the next recording, I'll basically go through how to analyze ELISA data. And it's important to know that when you are creating your standard curve, you need to be aware that the curve isn't linear. As I've said, it's this kind of S-shaped, this sigmoidal curve, where basically at really low concentrations or really high concentrations of the protein, the curve is definitely not linear. And you need to take that into account when preparing the standard curve in GraphPad Prism, but we'll go through that in great detail in the next session. If we move on to the next methodology, so this is multiplex assays and specifically flow cytometry. So multiplex assays are very similar in principle to ELISAs, and the main difference is that they use bead-based technology. And the big advantage of multiplex assays over ELISAs is it allows you to detect different proteins simultaneously. So what do I mean by that? Well, in a standard ELISA format, you'll only be able to really use one antibody that will be specific for one protein, and you'll only be able to detect that one protein in that clinical sample. In the microarray ELISA format, using specific beads, you're able to actually detect multiple different types of proteins within your clinical sample. And this is just a diagram highlighting the basics of this. So you'll have a fluorescent bead that is attached to a capture antibody. Capture antibody is specific for the protein in the exact same way an ELISA capture antibody is. That protein, if it's present, will obviously bind to the capture antibody. You can then use a detection antibody and also a protein. And instead of it being an enzyme, it's a protein that's attached to a fluorescent probe. And you're then able to detect, using flow cytometry, which we'll talk about in the next slide, the presence or absence and the quantification of different types of proteins within your clinical sample. So this is just a diagram showing the premise of this flow cytometry multiplex immunoassay. So you have your antibody attached to your beads that are specific for a given protein. You'll add your proteins or your standards. If the protein is present, then it'll of course attach to the antibody that's on the bead. You will then add a protein that's attached to a fluorescent probe. And if that fluorescent probe, if your original protein is there, then the detection antibody will of course be there, and then this protein with the fluorescent probe will be able to attach, and then you're able to use flow cytometry to detect the presence or absence and also the concentration of that protein within your sample. And this on the right hand side just gives you a brief or a very basic schematic of what flow cytometry is. Hopefully it's something that we've covered before, I think, in journal club publications, and publications have used this technology. But basically flow cytometry allows you to detect whether it be stained cells or in this case stained protein suspensions, and detect whether they are there or not. And basically what will happen is that these proteins, these beads, will pass through the laser. The laser light source will basically excite those probes if they are present within your sample, and those probes following excitation will release a certain light at a certain wavelength that can be detected by the machine. And this is essentially the basic premise of the multiplex immunoassay. Very similar principle to ELISA's, but has quite a lot of more advantages to this technique than ELISA's do. However, it is quite expensive to run this technique. So if you are looking for a wide range of different proteins, then absolutely multiplex immunoassays are a good option. If you're only looking for a certain handful, maybe 2 or 3 different types of inflammatory markers, for example, inflammatory proteins, then I don't think multiplex is the way forward. I would say ELISA's are probably your best option. So if we just move on into the last two, so we've got protein immunofluorescence and immunohistochemistry. So immunofluorescence is a type of immunoassay that uses a detector antibody labeled with fluorescent dyes, as you can see here in the diagram on the bottom right. Immunohistochemistry, on the other hand, is also used to detect proteins, but this involves a colorimetric change following the addition of a substrate. Very similar principle to ELISA's, as you can see on the left-hand side, but this is actually using whole tissue. So tissue biopsies, you can actually detect the presence or absence of a certain protein. So both techniques involve primary, and if you are using the indirect method of these methods, then it will involve a secondary antibody. So for immunofluorescence, this involves the principle of excitation and emission. So very similar principle to the multiplex immunoassays. So you have immunofluorescence here on the left-hand side. So if the protein is present within your tissue biopsy, then the primary antibody will bind to it. You can then add a secondary antibody that's conjugated to a fluorescent probe. And then following excitation, the fluorescent probe can then be essentially visualized using a fluorescence microscope. Usually confocal laser scanning microscopy is used for immunofluorescence. On the right-hand side, we have immunohistochemistry. But basically, if the protein is present, you'll have an antibody attaching. The primary antibody, the secondary antibody is conjugated to an enzyme, this horseradish peroxidase, for example. Once you add the substrate, the enzyme will catalyze the reaction in a similar manner to the process of an ELISA, and you get the production of a colored precipitate. This colored precipitate can then accumulate around the location of the antigen or the protein, and this can just be visualized using just normal microscopy, normal light microscopy. I've included these two images here from 2 papers from recent years, just to highlight the sort of nice images that you can generate from using these techniques. So on the left-hand side, I'm not going to go into too much detail on these. On the left-hand side, this is a paper from a few years ago, where the authors were looking at the importance of these proteins in maintaining tissue homeostasis at the oral mucosa. You can access this paper at this link here. But basically, it's just to highlight you can have different fluorescent probes that target different proteins within tissue biopsies. You can see here we've got a nice green, red, and blue stain. And when they're merged, you can get these nice colorful images. On the right-hand side, you have an example of immunohistochemistry. So this is a paper from a few years ago where the authors were looking at healthy gingiva and gingiva taken from patients with periodontal disease. And they were looking at these markers of hypoxia. So hypoxia is just very low levels of oxygen. And what they hypothesized was that patients with periodontal disease would be more hypoxic within their oral cavity, and therefore this should be able to be identifiable when they look at the tissue biopsies. And specifically using this protein, this HIF1 alpha protein, what they were able to do was stain these tissue biopsies for this protein. And if I zoom in, I don't know if it will allow me to zoom in, what you can see is this accumulation of these kind of these brown pigments, or these brown discoloration of the biopsy. And these brown pigments here are essentially the substrate of the immunohistochemistry process. So as we talked about in the last slide, you have a substrate, and in the presence of the protein, and thereby in the presence of the enzyme within a tissue biopsy, then the enzyme will cleave that protein, will cleave that substrate and change color. And therefore, what you get is this formation of this sort of brown discoloration around the cells that are positive for that protein, or in the presence of that protein within the tissue. And you can kind of see this, and the authors have highlighted that with these red hours here, and clearly you can see in the healthy gingiva there's no presence of this discoloration, and that is an indicator that this protein is indeed present within this tissue biopsy. So what I do want to emphasize before I move on is that these techniques, immunofluorescence and immunohistochemistry, is very qualitative in nature. And what I mean by that is that there's no way that you can quantify the level of expression of these proteins simply from just using these technologies. Generally speaking, when you use these technologies in a laboratory environment, or when you're trying to publish a research article, then these types of techniques are essentially used in line with quantitative techniques such as ELISAs, multiplex assays, Olink technology, which we'll talk about. So these images should not be used, these techniques should not be used solely without the help or the assistance of quantitative assays as well. So that's just something I want to make clear, is there's no way that you can accurately quantify the level of expression of these proteins without using other techniques, even though they do produce nice, pretty pictures. So finally, I just want to talk about what I've termed next generation platform technologies, and specifically Olink. So Olink uses technology similar to multiplex qPCR, and it actually combines ELISA and qPCR methodology. So I know we've not talked about multiplex qPCR, we've talked about multiplex flow cytometry, but basically this is the principles behind this technology, and this is known as proximity extension assay technology. And I'll upload a video or a link to YouTube with this recording. I won't play it in this session, but I will upload it as well if you are interested in going away and looking at what this technology is. But basically, what happens is that you have antibodies that are specific for the protein in a very similar manner to the ELISA protocol. But at the end of those antibodies, you will have nucleotide bases, so oligonucleotide bases attached to the end of the antibody. So if that protein of interest is present within your clinical sample, then you will have attachment of the antibody in the same way as you would in an ELISA. And then if that is the case, then you will have presence of these nucleotide bases, this kind of tag on the end of the antibody present within a given well. These sequences, these bases, will then be used as a template to basically extend and amplify that DNA. And as they extend and amplify that DNA, they use the normal qPCR method. So hopefully you've learned about that in the last session with Mark. And basically this extension and amplification of this will basically increase the amount of that signal within a given well. And it allows you to detect very low concentrations of a given protein simply because of this amplification, extension and amplification process. And that is one of the main advantages of Olink, is that it allows you to detect very low quantities of a given protein. So Olink technology provides many different types of panels of protein markers. So this is a company from Sweden, and I think there's also one of the sites is in the US as well. And basically what happens is you need to send off your samples to this company, and they will do the analysis for you. And to do that, they use a panel of different protein biomarkers. And these biomarkers and examples of these biomarkers can be essentially incorporated into a specific panel. And they already have pre-designed panels. So these pre-designed panels are just some examples here. There's many others as well. So things like the inflammation panel, the immune response panel. So basically, if there's any level of inflammation or elevated immune response within a given patient, then there are certain markers that you would be able to detect within the human body, within a clinical sample. They also have an oncology panel, so this would also be of interest if any of you are coming up with research proposals for a given oral cancer. And they also have a metabolism panel, which is involved, or which involves sort of key biological processes such as cellular metabolic processes, receptor signaling pathways, etc. And this technology can be used for most clinical samples. And on this right-hand side here, you can see an example of an inflammation panel. So I've used this technology quite recently for an in vitro experiment. And these are the 92 different proteins that they include in the inflammation panel. And it's quite a widespread amount of different proteins. So basically pro-inflammatory markers, anti-inflammatory markers, cytokines, chemokines, antimicrobial peptides, a vast array of different proteins that will allow you to determine, one, whether they are present within your clinical sample, or two, the actual quantity of them. And as I've said, I've quite recently used Olink for an in vitro study, and this is just an example of the data that you would get back. So don't worry too much about the treatment groups. I'm not going to go into detail about the actual experiment, but this is the data that you get back. Or should I say that you get the raw data back and you can use their software online to basically come to put together these images and these graphs. And this heat map here is an example. And what you can see on the bottom is all of the detectable proteins within my sample. So I think I found around 28 or 29 proteins that were actually above detectable limits within my samples. And what you're able to do is then group together the samples according to the expression or production of these proteins. And it kind of just highlights that, you know, this technology is really good because it gives you a widespread screening, if you like, of different types of proteins. So if you don't know exactly what the inflammatory response is going to be doing in a certain disease, or you maybe want to have a look at a vast array of different proteins, then Olink technology is really a very good piece of technology to use for that. It is expensive, very expensive, and therefore there are obvious limitations, and there's limitations to sort of the more standard technologies such as ELISAs or multiplex. It's a lot more expensive, but again, it would probably give you a lot more data. But it is very, as always, it's very much dependent on the research question that you are coming up with, and of course, what disease and what clinical sample that you're going to be working with. So that brings us to a close. I've gone a bit over on time. I was hoping this would be about 30 minutes, so I'm sorry, it's about 45 minutes in length. You shouldn't, after this session, you should now understand what inflammatory biomarkers are and have some examples from the oral cavity, which should hopefully guide you if you are interested in the immunological side of things when you're coming up with your research proposal. There's different methodologies. We've discussed some in a bit of detail, and obviously we've discussed the ELISA protocols in a lot more detail. Different methodologies and techniques can be used to assess protein expression and production within a clinical sample. And as always, as I've said, it very much depends on your research proposal. So you need to consider what oral disease that you are interested in, what clinical sample you would take to assess the inflammatory response within that oral disease, and then also which technique or techniques would you use. So of course you need a quantitative measure or quantitative assay, whether it be ELISAs, multiplex, or Olink, and you could possibly, depending on the disease, take or use have a qualitative methodology as well, such as immunofluorescence or immunohistochemistry, to get some nice, pretty images that go alongside your quantitative data. And you also need to consider other things, so cost of experiments, so essentially limitations, advantages, disadvantages of each of the techniques. And of course, each of the techniques has an advantage over another one, but it also has a disadvantage over another one. And those are the things that you need to consider when coming up with a research proposal. So that brings us to an end for the session. Please feel free to message me, email me, or use Microsoft Teams to send me a message if there's anything that is unclear in this session. I will upload this to Moodle. It's currently Tuesday afternoon, so I will upload this today or later on today. And then I will likely record the ELISA example. So the data analysis for this part of the session will be to go through, process ELISA data, and then come up with a standard curve, which will allow you to determine protein concentrations of your clinical sample from that standard curve. So I will upload that video probably Wednesday afternoon. And if anyone does have any questions, feel free to either message me or ask me in the lecture that I have with you tomorrow. So thank you for your attention, and I will see you next time. And I will specifically see you next week for the actual live session where we're going to go into detail on the data analysis. So thank you, and I will see you next time.